my training in bioinformatics. So in the last session, we discussed a lot of things together. We discussed the resources, and we saw that many of you have been able to, log in to different platforms. So today, again, we will be showing you the overview of how to access the resources, as many of the participants have, contacted us and asked to, understand how the resources can be accessed and how they can complete the courses and use the TBioInfo platform. So been just waiting for a few more people to join. So people are still joining. So we'll just start the session in a minute. Okay. So yeah. So in this program, as we have as we discussed last time, we will be covering some of the important topics and give you an overview of the program. So some of the topics that we thought that we will be covering during this program that would give you an overview to utilize the platform for various purposes, like research, different research from different areas of analysis that you could be implementing this platform for. So we will be discussing how to utilize the platform and how to utilize the resources that are associated with the platform. So over the years, I mean, we have been generating a lot of resources to simplify bioinformatics, and to do that, we have been developing these courses and projects. So the courses that we discussed last time, the courses give you an overview of how to utilize the data sets that have been published in reputed journals, and also has been well digested by the scientific community. So they are peer reviewed journals, and the data sets are available publicly, so that you understand how the study has been conducted, what experiments they have done, and also understand the outcome of the study, and how to look into those data sets, how they look actually, what they mean, and how to interpret such data. So a few of the topics that we will be covering includes the analysis of high throughput omics data. So last time we talked about different omics data sets that are there on the platform. So we discussed about transcriptomics, we discussed about metagenomics, we discussed about mutation variants, so genomics. So we also gave you an overview of how to start accessing the platform. For example, logging into the platform and then looking into the demo pipelines and how the demo pipelines work. So today, again, we will be going through the platform, and we'll be showing you some of the important aspects that, or some of the important resources or ways to understand the data type and also understand the research behind it. Again, I mean, utilizing these resources towards a research goal requires use of statistics, because we are talking about big data here, we are talking about data here. So in the end, what we are trying to look into is use of statistics to interpret or understand the patterns from the data. So we'll be using biostatistical tools and machine learning concepts during this program to understand how these technologies utilize machine learning concepts to make a significant observations or come out with significant results. And another aspect is to understand what this actually means in terms of biology. So first we're talking about data, then we talked about the techniques, then we talked about the advanced techniques like machine learning, And then from a machine, which the results that you get, how do you, biologically make sense of that result? How do you connect the result that you got from machine learning to a biological interpretation is something that is well covered in those courses, and also the examples that are given in those pipelines. So combining all these concepts together, I mean, gives you a holistic picture of what is happening. So we are conducting a research which has different components, like different multi-omics component, and how do you connect that and see a complete picture out of these techniques that have been, used to generate such data. So that is multi-omics integration, where we use, again, machine learning tools to interpret such complex data. So I think one of the outcomes that we think out of this kind of learning is to be able to, come up with some of your ideas, and propose a project, propose a research goal, an objective that you want to, work on, and that you want to actually, translate. So I think this gives you an opportunity to use the platform in the next step after learning, of course, and after practicing, because those are very important, components of doing a research, because you need to have that practice and confidence going into your own working into your own data, and coming out with, confidently with results that connects the data and also the biology. So one of the objective would be to, work on your own thing, and or maybe work on the projects that we work on, and complete this research journey. So we discussed last time a couple of resources, such as the server platform, and also the educational platform, and how they connect. And today also we'll be going through some of the concepts using both of the platforms. And I think a couple of the participants, or maybe few of them, have yet to receive the platform, the Bioinfo platform password and access details. So I think we will be sharing them very soon, maybe now. So please let us know if you still have to get the access. If you get it now, then also we can, do the hands-on together. So now I will pass it on to Ilya to take you to the organization page, which we discussed last time, to see the progress and the see the forum and what all resources that we have. Ilya, you there?
Yes. Okay. Maybe you can stop sharing the screen so I can share mine. Okay, thank you. Okay, welcome everyone. Yes, I wanted to just make sure that everybody has access to the course materials, and we'll just do a quick review of where to find all of this. So here's the link to our organizational page. So let's go to that link. Let me share my screen here in just a second. And what we'll do is just check while we are looking at the screen that everybody has set up their account. Everybody knows how to find the discussion. As you can see down here, we have activity of the different participants, so you can really see what people are working on and where they are in terms of their courses. We can comment on this activity tab. So if I see a pipeline, I typically go and check that pipeline just to make sure that this pipeline was created in an appropriate way and kind of makes sense, so that we're not just running pipelines that are not going to produce any results. And if I see something that will have a problem, I will typically comment on it. Here, if you have any general questions that you would like to have addressed in the next session, we have a forum specifically designed for these kinds of questions and topics. We also have project submission guidelines. One of the objectives, as Mohit mentioned, is to have everyone here participate in a project or develop a project of their own. So we'll be posting here links to the form where you can start submitting your project ideas. We will also be putting here some additional guidelines on how to prepare, how to find the right data sets, which is something that we'll do today, how to process and prepare your analysis project, and then how to visualize and present the results of your analysis. We will also highlight here the top performance in the program so that you can take a look at how others are working through the materials. Here we have a tab of courses. And so all of the materials that we will be covering today and we'll be covering throughout the rest of the program, we will be referring you to the different courses available on the platform. As you explore these courses, please do remember that there is a finish course button, and that is an important part of being able to register the points for completing a course. There are some assignments, practical assignments in the courses that we will take a look at in our next session. But for now, we did ask everybody to complete Introduction to Bioinformatics, and indeed we see that a lot of you have. And if you want to understand how do we think about the organization of these courses in terms of a syllabus, there's also a tab right here where you can go to the syllabus and see the session topics and how they correlate with some of the materials in these online asynchronous courses that you will do independently. So for example, last time we covered an overview, an introduction, the platform, the different resources, and then we asked everybody to work on Introduction to Bioinformatics. In today's session, we will talk about different types of data, where to find the data and different formats of the data, and show you how to utilize that data on the platform. And you'll see that down here we have Introduction to Genomics. Then we'll talk about processing data. How do we identify mutation variants? How do we quantify gene expression? How do we understand diversity of different microbial communities? And we'll talk about materials that are featured in Transcriptomics 1, Introduction to Metagenomics, and Genomics 1. So you can really look through the syllabus and see how the materials correlate with the topics that we will be covering. You'll also see that there's a progress tab. The progress tab, as we mentioned last time, is where we can monitor the overall progress of the whole group. So we can see the difference between educational activity and practical activity. We expect everybody to be at a certain level of readiness before we start working on novel independent project ideas. And so it's important for us to make sure that there is some kind of a balance between learning the materials that are online, which are going to be the yellow points here, and practicing how to apply some of those techniques to a data set that we have already prepared until you are familiar enough with the platform and with the tools to apply them to a project of your own. So these are some of those organizational materials. The recordings for each session are also posted on the forum. So you can go through the recordings of previous sessions. That is already posted. The previous session has been posted. And today's session will be posted soon after the session is over. So at this point, I just wanted to make sure that all of you have seen a lot of these resources. Beepsha was just emailing some of the remaining accounts this morning to you if you didn't get access to the server. And I wanted to just make sure that we have addressed some of the urgent questions that you might have for today's session. So I do see in the chat. Okay. So for those of you that missed the last session, I just want to show quickly where do you find the recording for the last session. And also, if you could please check your access to the server. The way you check is you hover over this icon and you should see the name of your account. Okay, when can we start using the server for practicing? Today. So please make sure that you have this login. No, if you have already taken those courses, that's excellent. You do not need to take them again. We will be showing you more of the developing your own project and working on a new data set part starting from today. So if you have taken some of those courses, that's excellent. You don't have to take them again. Okay, so recordings, session recordings right here. You'll be able to see the previous videos of the previous sessions. And if you can all check that your account is ready. And some of you had questions about, okay, well, how do I use this and where do I find the data, right? So that's some of the things that we will cover in today's session. Okay, if there's no other questions, urgent questions, let me start giving you a review of what we'll do today. So we have these two platforms, the educational portal where we have educational materials and projects that you can start working on. We have the server platform. And so those are all connected by this t-bio.info domain name. So we have the main t-bio.info that gives you an overview, educational side, the server side. So today we will talk about how do we access some of the tools and how do we apply those tools to some of the data sets that we can find online on NCBI, on TCGA, on different types of sections. I want to make sure that we understand what types of data go into which section. So for example, in transcriptomics, in this section right here, we will be using primarily FASTQ files. So these are raw data coming out of the sequencer. We'll talk a lot more about the process of sequencing and why the data has to be processed in very specific ways before we can analyze it. But what we need to understand today is that as opposed to a demo where you don't really need to upload any new data, here you have to start thinking about what is the type of data that you have, and there are multiple formats like FASTA, FASTQ, SAM, and BAM. And so we'll see how to find those types of data sets and how to upload them in here. The objective of these processing sections is to generate structured data. That structured data is going to be analyzed by other sections on the platform. So here, for example, we can quantify gene and isoform expression for all the present genes and isoforms. And then in other sections, you will be able to analyze that gene and isoform expression. Then in the educational section, we also want to make sure that you understand how the educational section correlates with the platform. So as you saw on the platform, you will have different tools and different examples. And here we will describe these examples. So this is a view from the epigenetics. We have a section on analyzing ChIP-seq and whole genome bisulfite sequencing data. So this course is going to explain and provide some examples on how to analyze data from those sections. On the platform itself, whenever you go to upload the data, you'll have to upload data from the sequencer, but also you will be asked to specify the reference genome. Some of those genomes are already uploaded, and some of those genomes you have to find again on NCBI or another source and upload those there. So we'll talk a little bit more about some of those data formats that you have to take to upload there. And then once the data is structured, you can use these different sections for data mining and annotation. And some of those sections you work with structured data. So those are going to be primarily tables in TSV and TXT format. And I'll give you an example of where you can find some of those and what you have to do to start using some of that data that is publicly available. For example, you can upload a table of expression and then simplify that table of expression to a level of sample variability, like we see here in this principal component analysis example. or develop a clustering, so identify groups of samples and how those groups of samples are related to each other. So we'll talk about, so all of these methods are for structured data. In other words, they are tables. Okay, so does anyone have any questions about accessing the platform? And has anyone already accessed the platform and tried one of the practice pipelines described in one of the courses? I just want to have a sense of how many of you have already done that. Okay, great. So someone has already started a practice pipeline. How long will a pipeline run? It depends on a lot of things. It depends on the data, the size of that data. For example, a typical transcriptomic pipeline might take a number of hours to run. Pipeline that works with tables is going to run maybe 5 to 10 minutes, depending on the complexity of the analysis. And it also depends on how many people are trying to access the same set of jobs at the same time. Okay, so has anyone except for Maya has anyone else run a practice pipeline? Maya, yes. You do not have to keep the tab open. You start the pipeline, and it's important. Sometimes people forget to press the button to start the pipeline. Before you start the pipeline, you just build the pipeline. You don't actually start it. But once you press that button start, you can leave, and that pipeline will keep running. You'll get an email notification once the pipeline has finished. Okay, great. So Jay ran 2 genomics 1 pipelines for the somatic mutations. Yeah, so those might take a little bit of time because those files are fairly large. All right, great. So 2 pipelines. Has anyone else tried running pipelines? Okay, great. Okay, so yes, we'll talk today about those SVL files. So it will be a good time to practice. And we'll make sure that everybody can understand where to fit in those SVL files and what exactly is inside. Okay, anyone else? All right, great. So the majority of you, it sounds like, are still getting started and getting oriented. We hope that by next time, everybody has sufficient experience with working with these different types of data sets so that you can participate actively hands-on in analysis that we'll discuss next time. The topic of today's session is the different types of data. We'll talk today about finding different types of sequencing data on Sequence Read Archive, what's called SRA. We'll talk about finding processed prepared tables of expression, for example, on the Gene Expression Omnibus. We'll talk about RefSeq and Genome Bank and FASTA files. How can we find reference genomes? And we'll talk about other types of repositories that are specific to a condition. For example, the TCGA, which is the Cancer Genome Atlas, where you can find a lot of multi-omics data on different types of cancers and different types of patients. And we'll talk also about NCBI Virus, a repository of data sets dedicated to viral sequences, where you can find annotated sequences, quickly navigate through them, select the ones that you're interested in, and even do some preliminary analysis to understand which ones are useful for in-depth analysis that you can perform on the platform. So we'll also think about how preparing or finding those data sets. If you want to run a pipeline, what are the things that you need to have to be able to actually start that pipeline in a successful way? Okay, so let's start talking about the big data issue in general. The problem with big data actually started happening around the 90s, where a lot of different types of data became more and more available and accessible to the public. And we've seen over the years a tremendous growth of these different types of omics data types but they also have to do with how those omics data types are annotated. So essentially, any sample has to have an annotation, otherwise it's not clear how we can utilize it. So now we have this problem with a lot of available data, data that has different types of annotations. The data is structured and prepared in different ways. And one of the biggest challenges is to be able to integrate and extract insights that are relevant in some way to some research problem or a clinical problem, especially in the biomedical domain. So what we are going to talk about in this program is how, and in this session in particular, is how to find some of those project ideas and project data sets. And we'll show you some examples on the platform that can guide you in a step-by-step fashion on finding those specific data sets and understanding what they might be used for and how to find the proper annotation for those. Specifically, a lot of this data is available on the different aspects of the National Center for Biotechnology Information, on NCBI. So NCBI is a US-funded project funded by the NIH, where a lot of this data is required to be deposited in different formats, specifically for referencing in publications and also for having public access to some of those data sets. Now, some of those data sets are going to be restricted. Some of those data sets are going to be validated. And so there's a lot of variety in how those different data sets have been checked, processed, or what kind of access you get to them. So we'll do a little brief overview today. You can also see that there has been a tremendous growth in the number of annotated genomes. And that is also something to consider. For different types of organisms, there are more or less studied genomes, and that means more or less known genes and their functions and pathways. And so we often have to think about that part of it as well when we have a question that we want to study with data. For example, with the potato project, the crop drought response project that we discussed, what Zani was mentioning, one of the challenges with the potato crop is that those genomes are not well annotated. In other words, there are a lot of genes that their function in potato is not known. It is known that similar genes, for example, from Arabidopsis thaliana and other model genomes, have been annotated. So a lot of times we have to really think how to find the proper data for us to be able to analyze that kind of data efficiently. So the first section that we want to go to is NCBI. So let's all go to NCBI together, and we'll try to find some of the possible data sets that you can analyze to see which section of the platform or what type of analysis might be best used for these different types of sections. So we'll go to NCBI, which is NCBI link right here. You will see here how there are many different databases available. And each one of these is going to contain different type of data sets that you can download or use, or different types of collections of data sets that you can see samples in. So let's say we are interested in breast cancer metastasis. Once you search for that, you'll see how there's a lot of different types of results. So today we want to look specifically at two examples. One example are gene expression omnibus datasets, right? So if you go to gene expression omnibus datasets, you see that there's a lot of them available. Now you can see where we can find different types of processed gene expression datasets and the different platforms that they have been prepared with. For example, if I'm interested in analyzing gene expression and isoform expression generated by Illumina platform, I can go to expression profiling by high throughput sequencing. And I can also go into maybe the recent year and select here. And you'll see that a lot of them are going to have uh, more or less informative titles. And you'll see that a lot of them will have different types of organisms. So for example, human, mouse, rat, etc. So let's look at human. Okay, so this looks like an interesting project. Let's take a look at what is in here. So you can see that the expression profiling by high throughput sequencing, the goal of the study was to identify temporal changes in breast cancer cells as they adapt to the brain metastatic microenvironment. And these are some specific type of brain, human breast cancer cells were injected into the carotid artery of mice and dissected at 7 or 40 days post-injection for RNA sequencing. So this is RNA sequencing, in other words, gene and isoform expression. Now let's see where the actual data is. So you can see that there are 12 samples. Those samples are organized by different days, and each day has multiple replicas. So essentially we have 4 different groups of samples organized by the day that they were taken. And down here, you'll see that there are multiple different formats that you can download. And down here, you have the full supplementary file that you can download. You'll also see down here a link to the SRA run selector. So what are these files? These files are processed. So in other words, they're organized into a table of expression. If you go down here and download this sample or this file, let's take a look at what's inside. You'll see all of the samples in TXT format. So for example, if we open this in Excel, you can see already from this view right here that these are names of genes and the level of expression, right? So this is day 7, replicate 1, and then here you can see names of genes. So looking at this file and looking at this collection of individual samples, how do you think we can analyze this type of data? Okay, so briefly let me explain. Okay, let's see. Compare between the expression between the days, level of expression by heat map. Okay, good idea. So for example, if we want to do a comparison, right, that could be a differential gene expression pipeline. If we want to create a heat map that's just visualizing those genes. But essentially, before we do any of these ideas that you're proposing, we have to organize the samples into a single table of expression. So before I do anything, before I can just simply take those files, I have to combine the individual samples by column, right? And make sure that they all have the same gene ID. And the gene ID, the expression of that gene ID is going to be across different columns. Once I have a table like that that is already prepared, I need to make sure that the correct identification is placed in headers. And then I can actually process that data in the ways that you're describing. So right here, we have differential gene expression. If we have multiple factors in our example here, for example, we can have days as number 1 factor, and maybe there's also different types of locations or different growth rates, right? So that could be factor number 2. So then we can do multi-factor analysis of those relationships between gene expression values that we observe and the factors in our experiment design. If we want to do visualization, there are some utilities right here for visualization, for example, creating a heat map. But the important point here is that all of these analyses, the input is a properly prepared table of expression. And so we'll talk about how to properly prepare a table of expression in our next session. But what about knowing, right, if we see right here, these are genes. These are genes that have already been annotated, and we don't have to worry about annotation. But in a lot of cases, it's not just the level of gene expression that affects a certain biological condition. It's also the alternative splicing events, or potentially it's variation at the exon level. Or sometimes we are looking for non-mRNA expression. If this is total RNA, we can find in there microRNAs or other types of non-mRNA data. Where could we analyze that kind of data? Well, as we saw, this link at the bottom to the SRA files, or this link right here for the SRA collection, is going to give us a link to the sequencing data. So this is going to be non-structured data, and pay attention to how big these data sets are compared to what we saw here. So here, all of this collection is 1.2 megabytes. If we go in here, each sample is over 1 gigabyte and close maybe to 2 gigabytes, sometimes even over. So if I have here 12, we're talking about 24 gigabytes in total. If I want to take all of this data and reanalyze processing for example, different types of isoforms and different levels of exon expression, then I would have to download them all to my computer, upload them to the platform. Obviously, that is not an efficient way of dealing with files. So instead, we have a way on the platform. Let's just go to the platform, and I'll show you an example. We have in this section, where we don't have yet a prepared table of expression, a way to deal with different types of uploads. So as you see, last time we looked a little bit at the demo version so that we can talk about the different steps of analysis and different types of output. But today, let's start by talking about this section right here. So as we set up our analysis, we have to define what type of sequencing this was, what type of a reference genome we're going to use, and what type of input we're going to use for our analysis. In this case, we are talking about human tumor cells. So they are metastatic tumors. And so when we can select here our model genome GTF, which means a model genome, a human genome is a model genome. It's a reference model genome, and it has an annotation file. So that file is going to give us the beginning and the end of each gene or isoform or a genomic element. So if we select this, the second selection we need to make is whether these are single end or paired end reads when they are sequenced. We'll talk a lot more about this in our next session. So for now, we can forget about this. These are going to be SRA files, which are sequence read archive. If we go here, we can see that these are SRA files. So if you click on one of these, okay, you can see that the download is an SRR, which is a sequence read archive. And then inside that archive, you actually have two FASTQ files. If you actually download and expand that archive, you'll see that there are two types of files, a forward strand and a negative strand, paired end sequencing. You can also see, by the way, here that it says that the sequencing is paired, layout paired. Okay, so a question, how to know which type of reference F genome to select? Well, like I said, you can have a fully annotated model genome, right? So our model genome stands for model genomes, and the model genomes are uploaded right here. So all of these are model genomes. You don't have to then upload a genome. If there's not a genome that is available on the platform for your project, and you have to find that genome on your own, then you will select either one of these two options. One is just the genome file, which is typically going to be in a FASTA format, or an annotated genome, which will have two files. One is a FASTA file for the reference sequence, and another one is a gene transfer format, which is a format to store the beginning and the end of each element in that genome. For example, gene annotation, isoform annotation, exon annotation, etc. Does that answer your question, Trishla? Okay. Does the bacteria reference genome refers to all species genome of or specific genome? Okay, so bacteria reference genome is, yes, a collection of all bacterial genomes. Please don't use anything that you see here before you've thought about the analysis, and think of the outputs whenever you're planning your analysis. And that's an important highlight of today's session, is to really have a plan before you run and do your analysis, because you'll just be waiting for a long time. And if you're waiting for a long time and the output is not what you expected, then that's going to be lost time. So let's make sure that we plan the analysis, we prepare for the analysis, and Trishla, like you're asking, how do we add all the necessary items that we must have before that analysis is performed? So I'll get to that point in a second. Okay, so in our case, let's just see what we do here. So this is paired end human genome. So we already have that uploaded. We have two versions of the human genome. For example, Homo sapiens GRCH 38 and 37. So we'll use the latest one, which is 38. We'll change this to PE for paired end, and then we'll go to file upload. And so what I want to talk about briefly here are some of the options right here. As you can see, I can upload files directly from my computer. I can add files from link, or I can paste those links in here. I can upload a table from NCBI directly, which are going to be references to the files that we saw right here. Okay, so I can have a reference for all of these or just some of these files, and we'll show you in our next session how to create a table of references. But today, because of some of your questions and because of some of these topics that are going to expand in their complexity over the next few sessions, as all of you will be working on something a little bit different, let's just go with an overview of this process. So one of the things that you see here are SVL links. Some of you have already tried running a demo pipeline or ran a pipeline in one of the course examples. So let's focus on those just to see how those work. You can see that in those sessions, as you mentioned, there are a lot of SVL files provided, right? So let's find an SVL file and see what that looks like. Okay, so I have some data sets for you to all have for this practice. Sorry. And this will give us a good sense for finding a specific genome, like a non-model genome, and also seeing the different types of formats. In here, you can see EBV sample.svl, right? So this is an SVL file that a lot of times you will find inside one of these courses or one of the projects that we offer on the platform. What is an SVL? It's just a link to the folder structure on the server. In other words, these files are full SRA files, but they have been uploaded into the repository. And we want to use this for our analysis. Okay? And please pause for a second with questions that are not directly related with what I am showing now, because I think after I show what I'm showing today, you'll have a sense of how to manage different files. And then later on, as there are unique cases of a project related that you are going to be working on, we will discuss those in the context of that project. Okay, so, and I'll address some of your questions as I get to a pause here. I just want to make sure that we get through some of this initial material. So here we have an SRA file. I can copy this file, just do Command C, I'll copy it, and I'll paste it right here, right? So this is one way I can use an SRA file. So if I have just one file, it's easy to just put it in here. Let's go to group selection. You can see that it looks at this link and identifies it as a single sample. If I have multiple FASTQ files, for example, paired end reads could be two separate FASTQ files, then it will identify both of those FASTQ files as a single sample. In this case, it doesn't make sense for me to create groups. Why would I create groups? If I want to do differential expression and compare two different conditions, I would use differential expression. But in this case, we don't need to use any groups. Now my files are uploaded. The reference is set. I have a reference genome. I don't have to worry about a reference genome because the reference genome was selected from one of the genomes that I already had available on the platform. Now I can perform my analysis. So after I click on Start, okay. For example, I would like to map this on the reference genome. I can map it on the reference genome, and I can visualize what the mapping looks like. I would click right here, and then I would click on End, Save. It creates the pipeline. I have to name the pipeline. When I name the pipeline, please make sure to use some recognizable name and don't leave it blank because that will just create a long list of all the jobs that you have selected. So I will call this Ilya 1, 2, 3. And then you have to click on Run Pipeline. So do not leave it because it will just be prepared and it will be sitting there. So if I leave this right here and go back to areas of analysis, my pipeline is still there. You see it's prepared. And if I go back inside, I will be able to run the pipeline. You can see also the uploaded file that I just used, or you can run the pipeline. If I go back to my pipelines, I can also delete this pipeline. Okay. Scenario number 2. In the scenario number 2, okay, let's move away for a second from transcriptomics, which is a complex series of steps. Let's go to a simple pipeline. Let me show you what else you have inside. So you understand the logic of SVL, right? I hope that was clear. There are some other data sets available here. For example, there's a FASTA file. Let's take a look at what's inside. A FASTA file is essentially a sequence of nucleotides or amino acids, could be either one. It could contain one sample or it could contain multiple samples. What are we looking at here? Here we're looking at a specific complete genome of Zaire Ebola virus. And this is just the first genome. So if you search for this symbol, you'll see that that's the only genome included in here. This is the accession number of this genome. So if you want to find this genome on NCBI, you just take this accession number and you can find it there. What about some of these other ones? Let's take a look at this file. Again, I will search for this symbol. How many genomes do I have in here? How many genomes do you think I have here? 3. Excellent. So I have a FASTA file with 3 different genomes. Okay? So now inside, I have multiple groups, right? Multiple FASTA files with number of samples inside each one. I downloaded them from NCBI. So let's go back and let's go to a different section. Here we will go to this section, phylogenetic tree. So please let me know once you got there to phylogenetic tree. Okay, once you're there, just type in yes. I don't want to rush and go too fast so that everyone can do this together. Okay, anyone else? Great. Okay, once we get to 10, I'll continue. So we have 4. Anyone else? Anyone else? Going once, going twice? Yes. Okay, so now we have 5. All right, 6, 7. 3 more. 8. Which option? You go to areas of analysis and right here, phylogenetic tree. All right, great. I think we got to 10. So everyone, you go to server.tbioinfo and go to phylogenetic tree. Excellent. Okay. Here, what we will use are obviously not FASTQ files. They will be FASTA files. So we'll select FASTA. Click on this button. And let's upload those files directly. So you'll click on Add Files and go to that Dropbox. Please download that to your computer to use it. And I gave the link in the chat. Okay, what we'll upload here are these 3 files: Ebola virus 3 Gabon, Ebola virus 3 Liberia, and Ebola virus 3 UK 2014. Click on Choose. You can see that all the files are uploaded. And now we go to Group Selection. Because they are multiples of FASTA files inside each one, I'll actually create 3 groups, A, B, and C. And I will put the UK in one group, Liberia in another group, and Gabon in another group. Yes. Okay. So outgroup for framing. You can see that it says upload one GenBank file, another format. What is inside a GenBank file? Let's take a look. Here we have a .gb file, which is a GenBank file. We can again open it in a text editor just to see what's inside. Okay, so what's inside? A lot more information and also organization of this genome by gene, mRNA, CDS, etc. But what's important about this is that it has sequences in amino acids, as well as down here at the bottom in nucleotides. So now I have a connection between codons and these nucleotide sequences. So that's what I need to add here, is add this file. So I'll add this file, Ebola Zaire 1997.gb, and it will be used for framing of those amino acids. Don't worry about the concepts if you're not familiar with them. We will have plenty of time to review these types of data sets in the future, but for now, just to deal with these different types of files. You can see here that I can upload a reference virus genome. Actually, I'm not going to use evolutionary groups, and I will use the GenBank file as the reference genome. And now I can create a pipeline. Again, don't worry about creating this pipeline. I just wanted everybody to see how the different file formats are used in different sections of the platform. But if you want to, you can wait until the session that we have for multiple sequence alignment, and then we will do a multiple sequence alignment, prepare a multiple sequence alignment pipeline. As you can see, different options are available at different stages of your pipeline creation. And I can always go back by right-clicking on the same button. Okay, so you can see here I have a lot of options. Here, I don't have a lot of options. And if I go into here, again, I have all of these options available. So make sure that you plan your analysis and you have a sense of how these methods are combined together for whatever purpose you want to use them. Let me quickly complete this pipeline. Again, I complete the pipeline, I name it, and then I have to click on Run Pipeline. Okay? Any questions about how FASTQ files, FASTA files, SRA files are used? And as you're thinking about those questions, if you have questions on how to use some of these files, in the meantime, I will show Vidit how to use HTTP options. Okay, so if I have a collection of samples, previously I showed you how I can take one link, and you can see that the options are here: SVL, HTTP, and FTP. Now make sure that this is a link to the actual file. For example, if I go here, and actually if I go back a couple of steps, okay, so if I go right here, oops, sorry, not all runs. Ravi, you just right click. So your right click will go back to all of the to the previous step. Please try it and let me know. If I want to download the SRA experiments, I click right here, and here I can go to send to. I can generate a file, and here select run info. It will download a file to my computer, which is just in a table. Let's open that table in Excel. You will see inside there's a link to the SRA. Okay, so if I copy this link, I can use it right here. The problem is that this is not actually a link to a file. So it's not going to let me do anything. Why is that? Well, because NCBI does not store their samples in a file format. They store them in a database, and you have to use a specific script to access the actual SRA file. So to do that, you cannot directly paste here a link to HTTP. You have to use this button right here, and we'll show you next time how you can actually upload the samples that you need and run a transcriptomics pipeline. Okay, so I hope that the file formats for SRA, FASTA, FASTQ were explained. SVL is definitely the easiest. It will be useful for you to work with projects that are already on the platform. A lot of them will provide you with an SVL. Again, an SVL file might contain a lot of samples, just like here. You can see that these are SRA files, but unpacked. They have _1, _2, which means that these are pair and reads. Typically, you will be able to download an additional file that explains how these are organized and what is their annotation. In other words, you need to know what groups they're in, right? Or what do they really represent? So that file is contained in an SVL. You can also download raw data from NCBI and use that whole table to upload and run your analysis. Now there's another place where you can download FASTA files, and a good example is NCBI. On NCBI, there's a tremendous collection of different viral genomic sequences. For example, we did an analysis in April, and we saw that with COVID-19, there's at least a 4 to 5-fold increase in the number of genomic sequences for COVID-19. But there are a number of other viruses available there as well. So how do you access those types of FASTA files, select them, and prepare them for analysis? What we did with Ebola was done here. So let me show you. Let's go to NCBI Virus, which is this link right here, to show you how you can quickly find those types of sequences. Okay, so this is a link. So please go to this link, and here you can click on search by virus. Here, let's search for Ebola and wait just a little bit, and you can see that Ebola virus pops up right here. In here, you will see that there are two different types of sequences. There are RefSeq, and then there are standard FASTA files, or standard accession numbers. You can filter them and organize them, and that's exactly the process that we went through to prepare those sequences that you just uploaded to the previous pipeline. But you can go inside each one and see, for example, their full FASTA file or a GenBank file. You can just download the FASTA, which will look like this. Or you can go back to the GenBank, which is more details about the different genomic elements of the sequence. But typically in an analysis, you would like to download multiple files and do preliminary analysis and process them in some kind of a format that you can do downstream analysis on. So you can just simply select multiple ones and then go to download. And here you have a few options in the formats that you would like to download them in. Okay, another option that we had, okay, are there any questions about what types of files you would like to download here? Okay, how can I upload my test dataset RNA-seq generated in my lab and map it to a reference genome for RNA-seq analysis? Great question. So this program for the next couple of weeks will cover public domain data only. Public domain data that could be referenced, either it is already uploaded to the server or it is on NCBI or some kind of a public repository. The reason for that is because we are responsible for the data that you analyze. In other words, if you are going to find data from patients and that data is restricted, you have to submit certain certifications that you are capable of working with that kind of data. If you do any kind of analysis and publish it, right? And we have some part of that, we just need to have proper securities in place for you to be able to do that. And that all comes with a research license. For the educational license, that's why you have certain restrictions. You can only work with public domain data. You can only go through an approval before you actually do the full analysis. And that's why we work on this project preparation process so that you are ready to do the analysis in an effective way. Does that make sense, Abilas? All right, great. So in the meantime, if you are just trying to map a data set onto a reference genome and do a sample RNA-seq analysis, I suggest using a publicly available data set, just like what we did on NCBI, looking at some of your data that might be similar. So here, for example, you might go to SRA, which will contain those raw FASTQ files. And we will, and then you can create a project or think about a project. Okay, other public databases other than NCBI and TCGA. There are repositories of different types of data. And Trishla, if you could, okay. So there are multiple different types of repositories of different types of data, right? So there are data sets for, again, FASTQ files, right? So a good example would be EBL. They have, sorry, EBI, EMBL, EBI, right? So they search, they have a repository of data sets that you have. Typically, a lot of them are also on NCBI, right? So there's a lot of overlap, but some data sets are available here and not available there. There are similar ones, and we actually have, depending on what kind of a topic, right? For example, cancer, virology, neuroscience. There are many different repositories, and typically they're going to be organized by a specific type of projects. So I want to give you a couple of collections where you can find some examples. So here is a collection of repositories. If you look through here, there's a collection of different types of databases where you can find and download different types of data sets. So here you can take a look. And here's a collection of resources for cancer research. So if you're looking for RNA-seq data for cancer research, this is a good link. Please go through this one. All right. When and how do we submit project proposals? So let's go back to this organization. So summer online training program is right here. Okay, and here let's go to syllabus. So because we have a collection of different people with different levels of experience, somebody is completely new and is still trying to understand how the different, sorry, to log in, how to do some basic things, and others are much more advanced. It's important for us to make sure that everybody's on the same page. And so that's why we created these online courses and projects for you to really go at your own pace in between the sessions that we offer in person like this. So let's go back to the syllabus, and I'll kind of show you where you can find some of these resources that we're discussing. Okay, so and I suggest you go to this link. Okay, so here you see that last session we asked everybody to complete Introduction to Bioinformatics, which is very high level and not practical. It's just a conceptual course that allows us to learn about you and your background in the first form that you fill out, but also allows you to conceptually map out which areas of bioinformatics are going to be covered in the program. As we start with omics and big data today, we ask you to go through Introduction to Genomics. Introduction to Genomics gives you the basics of nucleotide sequences, chromosomes, different types of variations in DNA that you might expect. Again, it's not a practical course, and it's really optional for those of you that are familiar with this kind of information. Here's when we start to do practical analysis. So when we talk about next session, right? Today we just talked about different types of repositories, different types of data sets. In the next session, we'll actually run a few pipelines together where we will give you examples of a pipeline for processing next generation sequencing data in its different variations. So we'll talk about RNA-seq data. We'll talk about metagenomic data, which is diversity of microbiome of different bacterial species using 16S ribosomal RNA or using total RNA from these environmental samples. And we'll also talk about processing data from whole exome or whole genome sequencing, where we look at DNA variation, identifying variants, like somatic mutations in cancer. So we'll go through those step by step next time. But today, if you could start by going through Introduction to Genomics, if you already know that material, you can continue with Transcriptomics 1 and Genomics 1, and that will give you some sense of how those pipelines are processed. And if you have any questions or any kind of topics that you would like to additionally address in the next session, you can actually take a look at how to post that in the forum so that we're prepared to address those in detail in the next session. Okay, for Stephen, if you're a newbie in bioinformatics, is it possible to have a documented step-by-step approach of the processes you have been describing, something like an SOP? So today, we just looked at different file formats, and we have some basic review of the tools on the T-Bioinfo platform. So what I would like to give you is just a general overview of the T-Bioinfo platform, a short video that you can look at to see let me see, 1 second. Let me give you a video. Wadzani, when and how do we submit project proposals? Yes. So project proposals, I will show you in the forum. Okay, so here is an example of a video that gives you a broad overview of the platform. And here's an example of uploading the files from NCBI. And that, I think, describes what we did today. Okay, project submission guidelines, right here in the forum for Wadzani. So we think that it's important to have a sense of where everybody is at for the project status. We envision that some of you are more ready than others. And so what I wanted to prepare you for this is to start thinking about these important aspects of a project. So you obviously have to have a title of your project, right? That project title might be similar to a title of a publication. For example, how RNA-seq data could be analyzed to find differentially expressed isoforms in metastatic breast cancer. Okay, just a random title here. But then that's not enough, right? So just to have a title doesn't give us any sense of what the project might be. So then the next thing you got to do is write an abstract. The abstract is typically several sentences long that describe why what you're doing is important to do, how you're planning to do it more or less, and what is the significance of the outcome. What are you expecting as a result? Then you need to find the links to the data. So again, make sure that these are public domain data sets. If you are ready to work on your own independent data sets that you've generated at your lab, that's also okay. You will just have to buy a research license, but then you can still participate in the training sessions and get all of your questions answered if that is what you want to do. So that is something you can specify. And ideally, if you have a publicly available data set, please identify a reference publication so that we understand what type of analysis, what type of a condition, what type of data you're going to be working with, and we can make sure that that is going to be incorporated into your analysis that you plan. So the process will look like this. By the time we reach our third session, which is tomorrow or next time we meet, we will post here a link to submit this kind of information in a form, right? So it's not going to be available this week. It's going to be available next week. You will have a form link here. You click on that form. That form will also be emailed to you. And you can fill out that form and we can start the review process. By the time this program ends, you should have a clear sense of how to do the analysis that you wanted, and then also some of the questions that you can address as you start planning for that analysis. Okay, Ravi, just wondering whether you're going to show TCGA example today. Yes, I'm going to try to if we have enough time. Okay, are there any other questions about what we did so far? Okay, so what did we do? We did an overview of the platform, the different tools, the different types of data, data that is stored on the server, data that is stored in publicly available databases, and data that is inside projects, right? So a lot of the SVL files, the tables that we will discuss, they are already inside either coursework that we have on the platform or projects that are included in the platform. What are projects? Projects are exactly what we did now. They are samples taken from a publication, organized in a certain way, and then analyzed, and all of those steps of analysis are described to you. So essentially, you can reproduce partial analysis that was done in the publication. Okay. TCGA. So how to analyze TCGA data and what are some of the benefits to multi-omics? So the Cancer Genome Atlas contains a lot of very interesting data sets. These data sets are organized by different types of cancers. For example, here you can see the liver cancer, hepatocellular carcinoma. And for that specific condition, you have a lot of different patients with different risk conditions, with different phenotypes like age, stage of disease progression, etc. And those are typically referred to as clinical data. This is not true clinical data in the sense that you don't get access to all of the clinical data. These are extractions from the clinical data and are useful for organizing patients into groups. You also have multi-omics data about each individual sample. The important part of it is that most of the samples contain all of the different types of data, or at least there's a small group that contains all of them. They also are all developed, the data sets are processed and prepared in a very similar way. How can you find this data? And what type of downloads might you have to process that data in bulk? So first of all, a lot of the databases like TCGA and other variations of TCGA hosted by UCSD or by the MIT Broad Institute, you can find them in different types of formats. So I'm just going to briefly show you two examples. The UCSC repository, where you can search for detailed variation. You can see the GDC data portal, where actually all of the raw data is located, and also Firebrowse, right? So those are the three things that we will briefly discuss. So let's first go to the data portal. This is the link. So on this portal, make sure that you understand this warning right here that tells you that this is protected information, and you have to be sensitive with this type of data because, of course, these are real people, real patients, and so we typically want to use it once we already understand what we're going to try and do. So here you can see that there are 65 different projects from 67 different primary sites, over 80,000 cases, over 570 different thousand files. And so a lot of the data is already annotated. So how do you actually access the data? If you go right here into projects, you can see what type of data is available. You can see them by primary site, by the program, and TCGA is just one of the programs, but there are some other ones here as well. Disease type, data category, right? So let's say you want to look for RNA-seq data. So this will be transcriptome profiling. Here again, you have different projects. So let's select TCGA BRCA, which is the breast cancer. For breast cancer, you have over 1,000 cases and 33,000 over 33,000 files that you can download. You can see that the raw data is going to be protected. You can't actually download the raw FASTQ files, the SRA files, without getting proper certification. Annotated, I mean when you saw genes, right? So they actually extract gene level information from all of the samples and provide you with some kind of an interface to understand how that gene is expressed across different tissues, or how is that gene, what kinds of mutations are present in some of the cases, right? So that's what I mean by annotated. You see right here, annotation. Okay, so here you can see different types of data, transcriptome profiling, right? How many files? So you can click on the files, and the bad thing or the uncomfortable way about this specific portal, in my opinion, is that you actually have a lot of variation, and you have to first select certain types of samples. There's no way to just download all of them in a structured format, right? But essentially, you can see what these formats are. There's FPKM, TXT, so this is going to be a TSV, essentially, a tab delimited file for a specific sample, right? So, and you can go through and see that there's microRNA, there are different quantification techniques like HTC counts or FPKM. These are going to be important as we learn about processing the data sets and analyzing the data sets, because not all types of data formats are going to be appropriate. Okay, so another way to download these in bulk is a portal called Firebrowse. So if you go to firebrowse.org, this is a replica of the same data set, the TCGA data set. And here you can go to mRNA-seq. Sorry, first let's select BRCA. Okay, and here you can see the same number, 1097, and out of those, mRNA-seq is going to have this RNA-seq data from Illumina. But here you can download the full table. So for example, if I look for RSEM, isoforms normalized, RSEM genes, right, or gene expression. So if I download this file, it will actually contain all of the 1,000 plus cases. You can see it's about, I don't know how much, maybe, you know, a gigabyte of a file, of a text file, of a table, where I will have all of these different samples. Now, before you just do anything with that sample, you will have some kind of an ID for each one of your samples. You have to obviously have some way to annotate those samples to understand who these patients are and what conditions do they represent. And that's typically going to be in this clinical table. So if you look at these different variations, you'll be able to see what type of data is available, metadata is available for each one of your samples. Okay, let's take a look quickly at what kind of information is here. Okay, so here you see that by ID, and the ID is right here, those are the IDs of those patients, you can start seeing the different types of information that is available, right? So right here you can see pathological diagnosis, patient barcode, patient UUID, patient days to birth, etc. So this is all kinds of information. You can see that there's a lot of information that is not available for everybody, but you'll be able to search through this kind of a table to find those types of samples that you need by their ID, and then find the gene expression data in the other files that we are downloading right here. Okay, so this was TCGA. We looked at Firebrowse. We looked at, well, here is the annotation, good example of visual annotation per gene. So you can see here a gene, you can see how it's expressed, for example, in the liver hepatocellular carcinoma project. And here you can see the different exons of that gene. And then we looked at the data portal where you can find all of this data in an interactive way, but you have to first make those selections before you actually download the file. Are there any questions? And I have a few questions for you. So let's think about this together. In which database can we find a reference genome? Where do you think we can find a reference genome for some of these non-model organisms? Great, NCBI. What type of a file would that be? What would be a reference genome? GenBank, RefSeq, FASTA. Okay, great. So for RNA-seq, you would use a FASTA reference genome. For a genome of a virus, you would use a RefSeq or a GenBank. But essentially FASTA. FASTA, and if you have a GTF file, a gene transfer format file, that's a benefit as well. Okay, I saw some of the other questions. Let me get to that in a second. So where can we find tumor and blood control whole exome sequencing data to study somatic mutations? All right, great. So TCGA, again, what format would that be in? What format do you think we can download from there? TXT. Well, okay, so in reality, TXT would be a format for gene expression. Typically for mutation data, somatic mutations would be in a file called VCF, variant call file. And we'll talk about that as you go through Genomics 1, you'll see what a variant call file looks like. Okay, where can we find data on cell line gene expression to understand age-related degeneration of brain tissue? Where do you think we can find such data? Okay, NCBI. So let's think about this. Where would we find this data in a FASTQ format? Which portion of NCBI? Geo, okay, so gene expression omnibus. Great, so that's one of the places where we would find. What about this question? In which part of NCBI can I find sequence read archives from model animals with patient-derived tumor xenografts? All right, great. SRA, obviously it's sequence read archive, right? So that stands for SRA. All right, great. So I see that a lot of you are on point and already understand where to find your files. Okay, so here's a brief assignment. This is not required, but this is something to let you practice. So if you can start looking for some of those interesting data sets on NCBI and TCGA, these are great repositories, very rich repositories. As you start looking at the actual data sets, you'll find how the systems work. And if you will have specific questions for us to address, I will be happy to address those in the next session, just before we start processing that data. And that will also help you think about your project idea, right? If you have some kind of a vague project idea, you like to study cancer, or you like to study, for example, toxicity of some chemical, that's great, but that's not enough to prepare a project. So as you start looking at the specific data sets, references to the different publications about those data sets, the variety of different techniques to generate data sets, that will really help you narrow down and focus on a specific aspect of that broad topic that you might be interested in. And I also want to remind you that it's very important to continue to make progress with introductory coursework so that every time we meet again, the time in between is spent going through some of the materials that do break down some of the topics that we discuss here briefly in a much more detailed way, with a lot of quizzes to help you evaluate how well you know and understand the topics that are being discussed, and practical assignments that you can try to do, and then we can check, identify any issues or anything that's not clear, and really try to address those in sessions that we have like this. And finally, please complete your profile as much as possible. If you have links to your social media, we'd love to connect with you. We'd love for others to connect with each other and really work as a team as we make progress. I imagine a number of you will have very interesting projects, and others might be interested in joining your project and contribute to that project based on their skills and availability. Also, if you have any questions, post those in the forum. Don't wait until 10 minutes before the next session for random questions. It will really give us enough time to plan to address those questions in detail. In our next session, we'll talk about processing next generation sequencing data. So we'll talk a lot more in detail about FASTQ, FASTA, and GTF files in the context of next generation sequencing, and what types of techniques are used to actually generate the nucleotide sequences, how those different sequences could be studied from the nucleotide level and amino acid level, how we can do alignment of reads, and what are the different formats like single end or paired end reads. And we'll actually talk about the significance of variation inside those reads and how those are detected to be actual variants and not random technical issues that are caused by the instability of the process of next generation sequencing. We'll talk about the overview of RNA-seq. How do we quantify gene expression? What are the major steps to quantify gene expression? And so I want to remind you that a lot of this information is available. If you want to have a good background for joining that session, if you start on Transcriptomics 1, that's going to be great. For transcriptomics, we have a whole series. It's going to be transcriptomics 1, 2, 3, and 4 that goes in that process, right? So it's designed to guide you through some of the basics until more and more advanced types of analyses. We have another one on genomics that is also organized from introduction to more complex issues that are addressed in genomics 1 and 2. And we also have this course on metagenomics, another type of data, also next generation sequencing, very different type of analysis. When is the next session? So the next session is going to be on June 3rd at 10 a.m. What is the difference between GRCH 3738 and HG 18 and 19? There are the annotation, right? So they did a revision of how these different, the start positions, end positions of different genes, exons, typical transcripts, all of that information is located in the GTF file. I think you will find a lot more information on the internet if you search for it. I'm happy to share some links if you want as well. Okay, any other questions? Briefly, for those of you that are still here, these are some questions to think about the interface of the program. Okay, links I will be happy to post. Again, as you remember, we have a forum. In that forum, we'll be posting the recording. We have some online materials for you to work through, and I think those are great alternatives for an actual textbook. I think if you complete the online materials, that's like completing a textbook. Okay, Ravi, if you're not able to download a specific lecture, I'd love to know which lecture you're trying to download so that we can help you with that. Again, on the forum, we have here a section for recordings. So we'll also place there these slides and the references that we use under session recordings. Thank you again for joining today. This is all that we have, and I would be happy to continue and interact with you in between these sessions. Please ask me any questions on the forum, and I am looking forward to seeing you again next time.